With the recent Energy Independence and Security Act, global requirements for secure sources of energy have stimulated new public and private investment in algal biofuel research. According to studies conducted in the late 20th century, microalgae cultivated in open pond systems can yield more than 10 dry tons of biomass per acre per year. Such species have impressive scientific potential, lending mainly towards their less complex structure, fast growth rate, high oil content, and the ability to prevent new CO2 from entering the atmosphere. However, there are numerous factors that play critical roles in the gene expression of biofuel synthesizing cells in microalgae, including light and irradiance, temperature, nutrient concentration, and turbulence. I narrowed my study down to nutrient accessibility and determined the effects of CO2, nitrogen, phosphorus, and potassium, the latter two of which have not been extensively researched, on the biofuel synthesis of 4 species of green algae. A closed photobioreactor was constructed and 24 algal culture mediums were prepared, factoring in NPK amounts as necessary. The overall experiment was broken up into 2 phases, a CO2 pressure test and an NPK mass concentration test. The independent variables in both tests were the amounts of CO2 and NPK synthetically added, while the dependent variable was the amount of biofuel synthesized per culture. After 30 days of experimentation, it was generally proven that algae exposed to increased pressures of CO2 and a greater concentration of NPK produce greater quantities of biofuel. However, in certain species, some tests did show that phosphorus and potassium limitation may more effectively allow algae to sequester carbon dioxide and thus produce more biofuel. Further statistical analysis led to show that there was a 76% confidence the results were not due to chance. Based on this test, I learned that algae can definitely be a major solution in our current energy crisis. By understanding the effects of all variables on the genetic makeup of algal biofuel synthesizing cells, scientists can learn to effectively expedite the output of biomass from algae and help save the environment.